The field of longevity science has rapidly transitioned from speculative theory to a translational frontier, where discoveries at the molecular and cellular level are increasingly being tested in humans with the goal of extending health span, the years of life spent in good health, rather than merely lifespan. The promise lies in developing therapies that target the fundamental processes of aging itself, rather than treating diseases one by one. This shift from reactive to proactive medicine is unfolding across multiple avenues, including pharmacological interventions, microbiome modulation, and epigenetic reprogramming, each moving from bench to bedside through rigorous clinical trials. Among the most intensively studied anti-aging agents are senolytics, drugs that selectively eliminate senescent cells, which accumulate with age and contribute to chronic inflammation, tissue dysfunction, and degenerative disease. Preclinical work has shown that clearing these cells in mice extends both lifespan and health span. Early human trials, such as those involving the combination of dasatinib and quercetin, have already begun in patients with age-related conditions like idiopathic pulmonary fibrosis. These first-in-human applications demonstrate safety and modest benefits, but larger studies are needed to assess long-term effects and broader applicability. Another class of interventions receiving widespread attention includes metabolic modulators. Metformin, a drug long used to treat type 2 diabetes, is being repurposed as a geroprotective agent due to its ability to lower oxidative stress, improve mitochondrial function, and regulate insulin signaling, all pathways implicated in aging. The planned Targeting Aging with Metformin (TAME) trial aims to test whether metformin can delay the onset of multiple age-related diseases in non-diabetic older adults, a landmark effort to redefine how clinical trials evaluate aging itself as a treatable condition. Closely related are caloric restriction mimetics like rapamycin and resveratrol, which activate nutrient-sensing pathways such as mTOR and sirtuins, associated with cellular repair and longevity in animal models. Rapamycin, originally developed as an immunosuppressant, has extended lifespan in multiple species and is now under investigation for age-related immune decline, muscle loss, and cognitive impairment. Human trials are still in early stages but suggest that transient dosing may improve immune response in older adults without severe adverse effects. The gut microbiome is another emerging target, as mounting evidence shows that microbiota composition influences immune function, inflammation, and even neurodegeneration. Studies of centenarians consistently reveal unique microbiota signatures, including the enrichment of beneficial species like Akkermansia mucinifila and Christensenella. Fecal microbiota transplants, FMT, have already proven effective for treating recurrent C. difficile infections and are now being explored in the context of healthy aging. Pilot studies are investigating whether transferring a youthful or longevity-associated microbiome into older individuals can recalibrate inflammatory markers, metabolic function, or cognitive health. While challenges remain in standardizing FMT and understanding host microbiome compatibility, this area holds immense potential for personalized and non-invasive therapies. Perhaps the most futuristic approach lies in epigenetic reprogramming. This strategy aims to reset the biological age of cells by modulating the epigenome, the chemical instructions that govern gene expression without altering DNA sequences. Groundbreaking work using partial reprogramming with Yamanaka factors, e.g., October 4th, SOX2, KLF4, CMYC, in mice has shown reversal of age-related epigenetic marks and functional rejuvenation in tissues like the retina and muscle. While full reprogramming risks oncogenesis and loss of cellular identity, partial reprogramming offers a balance between renewal and stability. Human applications are in their infancy, but companies and academic centers are rapidly developing safer delivery methods, such as inducible gene therapy systems and small molecule alternatives. Early trials in eye diseases and skin aging may pave the way for broader applications targeting systemic aging. Beyond individual therapies, there is growing interest in combination interventions, guided by multiomics and AI-driven stratification. Aging is a multifactorial process, and no single intervention is likely to suffice. Trials are increasingly adopting biomarker-based endpoints like DNA methylation clocks, proteomic age scores, and metabolomic signatures to evaluate intervention effectiveness more sensitively than waiting for disease incidence. This shift enables smaller, shorter trials with actionable insights, accelerating the translation of basic science into real-world solutions. Importantly, regulatory frameworks are beginning to evolve alongside the science. Agencies like the FDA and EMA are now considering how to define and approve interventions aimed at aging itself, rather than only disease-specific indications. The eventual approval of an anti-aging indication would mark a seismic shift in medicine, validating the concept of preventive geroscience as a mainstream field. Challenges persist, especially in ensuring safety, long-term efficacy, and equitable access to these emerging therapies. Ethical questions also loom large. Who will benefit first? Will life extension exacerbate inequality? How should society allocate resources toward extending life versus improving its quality? These debates are vital but should not overshadow the extraordinary potential of longevity science to transform healthcare. The transition from bench to bedside is no longer speculative. It is underway, measured not just in patents and publications, but in the increasing number of clinical trials seeking to redefine the boundaries of human aging. With each step, the vision of a longer, healthier life, rooted in science, not fantasy, draws closer to reality.